Okay. Thanks, Blakely. And first of all, thanks to everyone for joining us today. I know it's coming to the end of the year, so we really appreciate that you're joining. So as you know, this is the winter seminar, although I think we need to say end of year seminar because both Luciana and I are in the Southern Hemisphere where it's really warm for us. It is the final Validate event of 2022. And I think you'll all agree that there's been a really exciting series of seminars, and we're ending on a high note today. So just to mention, we are recording, so please let Blakely know if it's an issue for you. And then, of course, we really encourage questions, and it would be great if you could do this by video, but you can also type them in the chat or just, of course, raise your hand if you have a question. So without further ado from me, I'm going to, with great pleasure, introduce our speaker for today. So Dr. Luciana Balboa is an associate researcher at the Institute of Experimental Medicine in Argentina. And her research interest is in deciphering molecular aspects of the human immune response to Mycobacterium tuberculosis. And her team is focused on antigen-presenting cells, specifically monocytes, dendritic cells, and macrophages, and specifically looks at the modulation of their activation programs by the pathogen. So today, she's going to be telling us about how her team has been unraveling the immunometabolic pathways that regulate leukocyte function and their impact on the control of TB. So Luciana, over to you. I'm really looking forward to your talk.
Well, thank you. Thank you so much. Thanks all people at Validate for giving me the opportunity to share.
Sorry.
Yeah, it's okay.
It's still.
Can you listen? Okay. Can
try
you
again.
listen to me?
Yep, we can hear you. We can hear you. That's a bit better.
Great. I just was saying that I really very grateful for this opportunity. So, you know, for us being at the end of the world is not that common to have these impressive opportunities to share our data. So I'm very happy for that. And also I would like to thank for the opportunity of being part of the program for early career researchers that Validate have, which is an impressive program. I'm very happy, and it was very useful for me, and also because it gave me the opportunity to meet outstanding young researchers. I can see some of them right now at the Zoom. So it really is a great experience. I encourage you to go on if you're an early career researcher also. So I'm going to start by presenting just me like this. Okay, so I'm gonna, as I said, have been saying, I'm an associate researcher working in Buenos Aires, and during the last years, I started my own line of research on the topic of immune metabolism of TB. And I would like to share with you some of the major findings that we got in this area, and also to provide you some still unpublished data that we have that I would like to have your feedback about it. So just as a brief introduction, maybe for some of you is not familiar with the disease, and just to be sure that we are all on the same page, I would like to start by emphasizing the fact that although tuberculosis is thought of as a disease of the past, it still remains one of the worst, of the deadliest worst infectious killers. So TB is caused by Mycobacterium tuberculosis that more often affects the lungs, and it can be spread from person to person through the air. So last year, more than 10 million people fell ill with TB worldwide, and 1.6 million died due to tuberculosis. And for instance, in my country, in Argentina, last year there were 40,000 new cases of tuberculosis, and as you can see, incidences during the last years have risen, which is quite alarming. So all this alarming data highlights the fact that we are still far from controlling this even ancient disease, and that more research is needed in order to further understand how we can improve our immune response in order to better control the disease. So M. tuberculosis can evade or modulate the host immune response, surviving within macrophages and establishing a persistent long-term infection. Such chronicity leads to an extensive metabolic remodeling in the pathogen, but also in the host. And during the last years, we have largely studied how different metabolic adaptations taking place within, for instance, macrophages can affect the outcome of the infection. And we focus especially on the accumulation of lipid bodies leading to the foamy phenotype. And in the more recent years, we also evaluated the impact on the glycolytic pathway of macrophages, and even more recently also on dendritic cells. And this is going to be the focus of my talk from now on. So it is widely accepted that metabolic pathways have a major impact on the effector functions of immune cells. And this is basically the concept of immunometabolism. And when talking about macrophages, the two more extreme phenotypes of macrophages that have been described, which are the M1 and M2 macrophages, are known to rely on the prevalence of different metabolic pathways that characterize each of them. So for instance, in the case of M1 macrophages, that are macrophages that are known to bear a high microbicidal activity, these macrophages are characterized by a metabolic change towards glycolysis and away from the mitochondrial respiration. And this metabolic switch is governed by the transcription factor HIF1 alpha, and I'm going to talk a lot about it today. So this transcription factor can potentiate the aerobic glycolytic pathway, leading to a high secretion of lactate, and can at some point allow the interruptions in different points of the Krebs cycles, leading to a lesser oxidative phosphorylation activity and a lesser mitochondrial respiration pathway. So this kind of this metabolic profile allows the cells to respond rapidly to the pathogens and also to produce antimicrobicidal compounds. On the other side, we have the M2 macrophages, which are known to be associated with healing and tissue reparation. And in this case, the metabolism that characterizes these macrophages is more dependent on mitochondrial functions, such as the oxidative phosphorylation and fatty acid oxidations. So these macrophages produce high amounts of ATP, but in a more delayed way than the M1 macrophages, but it's supposed that this delayed production of ATP is compatible with their longer term functions, which, as I say, are related more with the return to homeostasis and healing and so on. But in terms of the control of the infection by, for instance, Mycobacterium tuberculosis, M1 macrophages and their glycolytic capacity is associated with the control of Mycobacterium tuberculosis, and the M2 macrophages are more associated with the persistence of Mycobacterium tuberculosis. So we know that the immune response in TB patients is incomplete, and that's why we wonder whether the microenvironment that is established during the infection with Mycobacterium tuberculosis can at some point interfere with the metabolic network associated with the M1 profile. So in order to model or to test this question, what we did is to use pleural effusions from patients with TB as a physiologically relevant fluid capable at reflecting the microenvironment, which is found in the human cavity, in this case, the pleural cavity, and that is impacted by Mycobacterium tuberculosis infection. So basically what we did is to generate human macrophages to activate them or not towards the M1 profile by using classic activators in the presence or not of the pleural effusions from TB patients. And these fusions, we use them and we process them in a way that no detectable CFU were within them. So what we first observed is that, as expected, M1 macrophages, which are known to have a high glycolytic activity, can produce higher lactate in response to the M1 polarizing agents. But when we perform the activation in the presence of the pleural effusions from TB patients, the release of lactate is reduced. We observe the same effect even when we use different pools of pleural effusions from different patients, different group of patients, or even by using individual samples of single patients. So all of them, when they were associated with TB, all these fluids were able to reduce the release of lactate by M1 macrophages. But we didn't observe the same effect when we tested other fluids, other pleural effusions that were associated with different etiologies, such as heart failure, cancer, or pneumonia. So we also observed that the pleural effusions from TB patients could impair the expression of glycolytic genes, such as exokinase 2, lactate dehydrogenase A, and also the expression of HIF1 alpha, that, as I said before, is a key transcriptor factor that can promote the expression of glycolytic genes, was also reduced in these macrophages, M1 macrophages, that were generated in the presence of the pleural effusions from TB patients. So it was clear for us that the pleural effusions were inhibiting the aerobic glycolysis of M1 macrophages. But not only that, the pleural effusions were also promoting the mitochondrial respiration in these macrophages, something that is not natural in this kind of macrophages. And we observe an increase in the consumption of oxygen and the production of ATP associated with mitochondrial respiration in these macrophages, and M1 macrophages that were treated with the pleural effusions from TB patients. And we also observed that the morphology of mitochondria were different within these macrophages, and they were more characterized by having larger and more tubular mitochondria, which are compatible with their more bioenergetic status. So we also evaluated the impact of the treatment of macrophages, of M1 macrophages, with the exposure of these macrophages to the pleural effusions. We also evaluated the impact on pro-inflammatory functions displayed by these macrophages, and we observed that the release of IL-1 beta was reduced, and also the production of mitochondrial reactive oxygen species was reduced. But the bacillary loads associated to these macrophages that are exposed to the pleural effusions were increased. All of this suggesting that the pro-inflammatory and microbicidal capacity of these macrophages were at some point impaired by the exposure to the pleural effusions from TB patients. So we tried to revert all these inhibitory effects that was inherited by the pleural effusions from TB patients. And what we did is try to stabilize the expression of HIF1 alpha, that, as I say, can promote the glycolytic pathway, but also can promote pro-inflammatory features in the macrophages. So what we did is to use different drugs, such as DMAC or cobalt, that are known to inhibit those enzymes that normally degrade HIF1 alpha. So by doing so, we ended up by stabilizing the expression of HIF1 alpha and by increasing the levels, the secretion of lactate, than the glycolytic activity. So as you can see, when we have the M1 macrophages treated with the pleural effusions, the production of lactate is low, but we could revert it when we stabilize the expression of HIF1 alpha by using, for instance, DMAC or I'm going to try to change it, or cobalt. So we also were able to restore or to revert the other parameters that I mentioned before. The pro-inflammatory parameters are not showing them now due to the sake of time. But most importantly, we were able to improve the control of the infection in these macrophages that were exposed to the pleural effusions from TB patients, but in which HIF1 alpha was stabilized by using this drug. So our next step was really try to identify which was the nature of those components within the pleural effusions that were responsible for driving these metabolic alterations that we observed in human M1 macrophages. So to make a long story short, what we observed is that when we use the fraction derived from the pleural effusions that was enriched in lipids, is shown in green, when we use this fraction, we were able to reproduce the inhibition in the release of lactate.
Luciana, your microphone's got a bit strange again.
Sorry. Now?
That's okay.
Okay.
I think that's better. There we go.
I moved too much, sorry. I'm gonna just recapitulate a little bit. I was telling that we were trying to elucidate which was the nature of the component within the pleural effusions that could be responsible for driving all these alterations. And what we found is that when we use a fraction enriched in lipids derived from the pleural effusions from TB patients, which is shown in green, we observe the same effect, the inhibition in the release of lactate, and also the same effect on the rest of the parameters that I showed before. But even more, if we use a fraction that was enriched in a certain family of lipids, which are polyunsaturated fatty acids, we were able to reproduce the inhibition in the release of lactate, the inhibition in the release of IL-1 beta, the inhibition in the expression of HIF-1 alpha, and also the exacerbation in the vascular loads associated to M1 profile. So with all this data, we ended up by publishing this work in which we basically demonstrated that host-derived lipids within the pleural effusions from TB patients were able to inhibit the activity of HIF-1 alpha, leading to a lesser glycolytic and lesser microbicidal phenotype. But still, we wanted to know certain components that could be responsible for mediating this inhibitory effect on the metabolism and on the effector function on the microbicidal activity of macrophages. And that's why we decided to perform lipidomic analysis of pleural effusions from patients with TB and from non-TB patients also. So we detected the abundances of different polyunsaturated fatty acids within these samples, and we tried to perform correlationships between the abundance of each lipid mediator and the capacity of each pleural effusion to inhibit the release of lactate by M1 macrophages. So by doing so, we found 4 lipids, which are shown in red, that they show a positive correlation between their abundances within the pleural effusions and the capacity of each pleural effusion to inhibit.
Luciana, I think we lost you. Can't hear.
Yep, we lost
the glycolytic activity
of
yes.
You disappeared for about
now?
Disappeared for about 30 seconds. You're back now. I know.
I'm back.
Yes,
yeah.
We can hear you now.
Okay, sorry for that. I think it's my connection.
It was about 30 seconds.
Okay, so I'm gonna repeat this part. So I was telling that we tried to figure out which were the components within the pleural effusions. We knew so far that they were polyunsaturated fatty acids that could mediate the inhibition in the metabolism and on the microbicidal activity of macrophages, but we wanted to know which polyunsaturated fatty acids could be involved. So we performed lipidomic analysis of the pleural effusions from TB patients and from non-TB patients, and we performed correlationships between the abundances of these lipid mediators and the capacity of each pleural effusion to inhibit the release of lactate by M1 macrophages. So by doing so, we ended up by finding 4 lipids, which are shown in red, which are 7-myrisine-1, protecting the X, resolving the 5, and 18 HIP. These 4 lipids display a positive correlation between their abundances and the capacity of the fluids to inhibit the M1 glycolytic metabolism. So when we look at these lipids at the literature, because we were not experts on lipids, so we were like kind of surprised by finding these compounds. And we realized that the 4 of them were at some point related, and that they the four belong to the same family of lipids that are known as specialized pro-resolving mediators. So these lipids are compounds that can be made up by the body from omega-3 fatty acids, and the discovery of this family of lipids really transformed the way in which we understand pro-resolution, because for a long time it was thought that pro-resolution was only a mere antagonistic response to inflammation. But now we know that pro-resolution is an active mechanism in which compounds such as these lipids have an active role by actively promoting the resolution. And in the case of our project, it really made sense for us that these compounds that were known to be associated or to display anti-inflammatory and pro-resolutive characteristics, that they could be involved in attenuating the effector functions of M1 macrophages. So we tested the ability of commercially available versions of these four lipids to inhibit in vitro the release of lactate by M1 macrophages, and we found that 2 out of 4, specifically 18-HIP and resolving D5, were able to reproduce the inhibition in the secretion of lactate at the same extent as the pleural effusions from TB patients. The same 2 lipids were able to inhibit the expression of HIF-1 alpha and also to reprogram the metabolism of M1 macrophages. So these macrophages, M1 macrophages that were exposed to resolving D5 or to 18-HIP, they were less glycolytic and they are more dependent on the mitochondrial respiration. These 2 lipids also reduce the release of IL-1 beta by these macrophages, and also promoted the bacillary loads associated to these macrophages. And we also were able to restore the capacity of these macrophages that were treated with the lipids to secrete lactate by using the MOC, meaning by stabilizing HIF-1 alpha expression. And we also restored the secretion of IL-1 beta and also the control of the infection. So omega-3 derived fatty acids, and in particular 18-HIP and resolving D5 that are present within the pleural effusions from TB patients, could impair the glycolytic activity of M1 macrophages, leading to a poor control of Mycobacterium tuberculosis infection. And as far as we're concerned, this is the first evidence for the role of these specialized pro-resolving lipid mediators in the pathogenesis of TB. So we were also very interested about evaluating the impact of the microenvironment of TB, but also on other associated infections. And of course, we thought of HIV due to the fact that these 2 pathogens are very frequently associated, and due to the fact that TB is the leading cause of death among people living with HIV. So in collaboration with our French partners from Toulouse, led by Olivier Nerols, Christelle Verolet, and Giancarlo Lugo Villarino, with whom we created an international associated lab, we try to understand how tuberculosis-associated microenvironment increase susceptibility to HIV infection. And I'm just going to provide you the main messages of these 2 articles that are already published. But basically what we demonstrated is that the microenvironment associated with tuberculosis can promote the generation and the formation of nanotubes in macrophages. And nanotubes are basically transient membrane projections that facilitate intercellular communications. And through these nanotubes, viral particles of HIV can be transferred from cell to cell. So providing an alternative explanation for the already known synergism between the 2 pathogens. And also in collaboration with Gabriel Duet and his supervisor, Sarah Palmer from the University of Sydney in Australia, we also have some preliminary data showing that the pleural effusions from TB patients can have also a detrimental effect on the functionality of CD8 T cells against CD4 HIV infected cells. So what we demonstrated is that the treatment of the exposure of these CD8 T cells to the pleural effusions could impair their killing capacity, their ability to eliminate CD4 HIV infected cells. And we also have data showing that there is a clear impact on the glycolytic activity of these CD8 T cells. So we are still working on this project, but I think kind of like improves our understanding on how the microenvironment associated to TB can not only impair the control of tuberculosis infection, but also of associated pathogens. So going back to our story in which we demonstrated that HIF-1 alpha stabilization was important for the antimicrobicidal activity of environment macrophages, we have also some interesting findings by trying to stabilize the expression of HIF-1 alpha by giving the mock to MTB infected mice. So by doing so, we observed that the bacillary loads in the lungs of these mice were lower. And we also observed that there was a metabolic shift in the alveolar macrophages of these MTB infected mice that were treated with the mock from an N2-like metabolic profile towards an M1-like metabolic profile, which is supposed to be more microbicidal and could at some point maybe explain the lower bacillary loads associated to the lungs of these mice. But we were interested to see that these effects occurred at very early times post-infection. So that's when we started to thought that maybe HIF-1 alpha can also have a role in the functionality of dendritic cells. So, and that's why maybe regulating the initiation and orchestration of the adaptive immune response, a process which is known to be delayed upon MTB infection. So that's how we started to wonder how HIF-1 alpha mediated glycolysis could influence dendritic cells activation in TB. And basically, we started by characterizing the metabolism of dendritic cells that were exposed to irradiated or viable Mycobacterium tuberculosis. And we observed that in both cases, either irradiated or viable, glycolysis was induced, as evidenced by the release of lactate, the uptake of glucose, the upregulation in the expression of HIF-1 alpha. And we also observed, we also have some evidence of the increase of the mitochondrial respiration pathway, given that we observe an increase in the mitochondrial mass of MTB-stimulated dendritic cells, and also the mitochondria were larger and bigger within these dendritic cells that were treated with Mycobacterium tuberculosis. In fact, we observed that by the Seahorse analysis, we observed that both pathways, the glycolytic and the mitochondrial respiration, were increased in response to Mycobacterium tuberculosis. But when we observe the related contribution of both pathways in terms of the production of ATP, we observed that there was a much more contribution of glycolysis when the cells were stimulated with Mycobacterium tuberculosis to overall ATP production than or in comparison to mitochondrial ATP production. So we also validate these results by using a single cell method to evaluate the metabolic dependency capacity, which is the CONICET method that was developed by Raphaël Arguello, also in France, but in Marseille. And what we observe is that the mitochondrial dependency of the dendritic cells that were stimulated with Mycobacterium tuberculosis were reduced, but the metabolic capacity, the glycolytic capacity of the cells was increased. So we conclude that there is a major related contribution of glycolysis to overall ATP production when the dendritic cells are stimulated to Mycobacterium tuberculosis. So we decided to evaluate what happened if we inhibit HIF-1 alpha in these MTB stimulated dendritic cells, and from now on we used the irradiated version of Mycobacterium tuberculosis, and we observed that the inhibition of HIF-1 alpha by using this specific inhibitor resulted in the reduction of the production of ATP associated to glycolysis without impairing the production of ATP associated to the mitochondrial respiration, and that the inhibition of HIF-1 alpha and thus the inhibition of the glycolysis has a slight impact on the acquisition of some co-stimulatory molecules. But we didn't observe any effect at all on the capacity of these dendritic cells that were in which HIF-1 alpha was inhibited or not to polarize different CD4TH cells. But for the dendritic cell to be able to activate T cells, it first has to migrate to the lymph nodes. That's why we decided to evaluate the effect of HIF-1 alpha on the migratory capacity of the dendritic cells. And we observed that when we inhibit HIF-1 alpha activity in these dendritic cells that were stimulated with radiated Mycobacterium tuberculosis, or when we inhibited directly the glycolytic pathway by using oximate, we were able to reduce the chemotactic activity of these dendritic cells against CCL21, which is a ligand that regulates the home into lymph nodes. And also we observe a decrease in the migration of these cells through collagen matrices. We also validated this result by using a murine model in which we transfer bone marrow derived dendritic cells that were stimulated or not with Mycobacterium tuberculosis in the presence or not of the inhibitors of HIF-1 alpha or the inhibitor of glycolysis. And we observed that we could recover lesser number of cells, of dendritic cells in the lymph nodes when the cells were treated with the inhibitor of HIF-1 alpha or the inhibitor of glycolysis. So we conclude that HIF-1 alpha mediated glycolysis is required to trigger the migration of MTB stimulated dendritic cells into lymph nodes. And then what we wanted really to see is what happened with those dendritic cells derived from monocytes from TB patients. So we decided to evaluate the chemotactic activity of these dendritic cells derived from monocytes from TB patients, and we observed that it was indeed reduced compared to the chemotactic activity of dendritic cells from healthy donors. And we observed that in parallel to this decreased chemotactic activity, these dendritic cells from TB patients also were not as good at producing lactate in response to Mycobacterium tuberculosis, and they didn't upregulate their glycolytic capacity. But when we treated the cells with DMOC, meaning that we stabilized HIF-1 alpha, we could at some point improve their chemotactic activity. So we were surprised by the fact that we observed still differences in the dendritic cells derived from monocytes from TB patients not healthy, given the fact that all the protocol for differentiation was exactly the same in both cases. So it means that the differences should be determined at the level of their precursors, meaning the monocytes. And that's why we decided to finally evaluate the metabolic profile of monocytes from TB patients compared to healthy donors. So we evaluated the glycolytic capacity of the main subpopulation of monocytes, and we observed that the CD16 positive subsets of monocytes display a higher glycolytic capacity compared to the same subsets when they were derived from healthy donors. And we were quite excited about these results since we have some long years ago, we have already described that the CD16 positive subsets of monocytes were increased in TB patients, that they correlate with the severity of the disease. And we also observed that these monocytes were at some point more prone to give rise to tolerogenic dendritic cells and immunosuppressive macrophages. So as a final slide, I just would like to try to provide you a kind of evidence in which we try to connect the metabolism of monocytes with their ulterior capacity to give rise to migrating dendritic cells. So what we did is to use monocytes from healthy donors, and we activated their glycolytic pathway during the first 24 hours of differentiation towards dendritic cells. And we demonstrated that this treatment during this first 24 hours resulted in the increase of the production of lactate in these monocytes. And thereafter, we evaluated the chemotactic capacity of the resulting dendritic cells at the end of the differentiation, when they were exposed or not to irradiating Mycobacterium tuberculosis. And what we observed is that the early activation of HIF-1 alpha and early activation of the glycolytic pathway during the first 24 hours of differentiation in the precursors leads to the generation of dendritic cells that display a poor migratory capacity against Mycobacterium tuberculosis. So having said all that, I just would like to give you the final take-home messages that summarize, or at least try to summarize everything that I say. First, that the omega-3 derived fatty acids from pleural effusions from TB patients can impair HIF-1 alpha mediated microbicidal activity in macrophages. That the pleural effusions can inhibit also the control of Mycobacterium tuberculosis, but also can promote HIV replication. That HIF-1 alpha activity is required to trigger the migration of M tuberculosis stimulated dendritic cells, and that the premature exacerbation of HIF-1 alpha mediated glycolysis in monocytes can lead to the generation of dendritic cells with a poor ability to migrate. So I hope I have provided you evidence to show that HIF-1 alpha mediated glycolysis plays a major role in macrophage microbicidal activity and also in dendritic cell migrations, and that its modulation appears as a promising tool to develop new strategies to manipulate the immune response. So I just would like to thank to all the people involved in the generation of these results, especially to my two PhD students, Mariano Mayo and Joaquina Barros, also to Marine Joly, which is a French physician that spent 6 months in our lab this year, to my French colleagues, especially Christelle and Olivier and Giancarlos, Zoe and Sarah, and Rafael Arguello, who is the one that developed the STANIT method to evaluate metabolism at a single cell level, to the physicians, to my Mexican colleagues, and also to Gabriel and Sarah from Australia. Thank you for your attention. And of course, I'm open for questions.
Thank you very much, Luciana. That was a really lovely talk. I'm just waiting to see if there are any hands. I'm not seeing any questions in the chat. There we go. The first one is there. So Sasha, if you're able to go on mic, please do so. Otherwise, I can read out your question. Okay, I will read it out for her. So says, thank you, Luciana, wonderful seminar. I love the human-derived data paired with preclinical modeling. Very compelling. There's some evidence that glycolysis in T cells, CD8 to my knowledge, controls translational regulation of interferon gamma. Do you know if there is similar evidence in macrophages? Apologies if she missed that point.
Okay, so thank you, Sasha, for the comment. We haven't studied so far that interesting concept that you mentioned. There is a clear like antagonism that is described in macrophages between glycolysis and but interferon type 1, not interferon gamma, and it's antagonic. In the case of interferon gamma, it's expected to increase glycolysis in macrophages because it's kind of the M1 profile that one expects for macrophages can be induced by interferon. In our hands, when we use only interferon, the induction of glycolysis was not as pronounced as when we combine interferon and LPS. But you mentioned in T cells, CD8, the translational regulation. We will really look further on that, because now that we are starting to work on the CD8 involvement on how the microenvironment can also have an impact on the CD8 effector function. So maybe that can be a good insight, because we clearly observe an impact on the glycolytic capacity of the CD8 cells in the context of MTB infection. So thank you.
Thanks for that question. Thanks for an insightful, inspirational presentation. Just waiting for some additional hands.
Yeah, there is someone that with the hand raised, but I don't know. I will pronounce it Pishu Lal.
Apologies, I'm not seeing the hands. Oh, there we go. Go ahead with your question, Piyush.
Piyush Lal.
Piyush.
Maybe go with another one, and I will message her now.
Okay, so I mean, maybe while we're waiting for Piyush to ask the question, or others to think. So Luciana, and again, I'm going to show my bias here, but I was wondering two things. So in your data where you've used the pleural effusions for simulation, and in other assays where you've used your irradiated MTB. So two questions about that. One is, have you looked at, is there an influence of disease stage on your readouts? And could the mycobacterial strain that you use influence your assays?
Question, you asked whether the, sorry,
so for the first question, would disease state of the patients that you get your pleural effusion from influence your measurements? So the stage of disease. So if someone is newly very high burden,
that's very important question.
Yeah. Sorry,
your mic is a bit bad again, Luciana. Your mic's gone strange again.
I hope now it's better. Concerning the clinical status of the patients, I have to say that the pleural effusions, the cohort of pleural effusions that we have are all representative of very advanced and severe disease. We don't have samples from, let's say, less severe patients in general. The patients that we have is patients that need to be and to underwent the functions, you know, the destruction of the pleural effusions, because they are having some respiratory issues, and even when they take that, they remove the fluid, we still sometimes don't know the etiology. We have to wait, but they arrive when the situation is out of control. Let's say so. They are having like already symptomatic signs, and I mean they are representative of people that is having the disease, and they are in the moment in which they are showing symptoms. And about the lab strain that we use, for most of the assays we use the reference lab strain, the 837 RD, but we have another line of research, and I didn't have time to show the results, in which we try to compare different clinical isolates, how they can really differ or not on their ability to induce, to trigger glycolysis in macrophages. and how it can be related with their capacity, with their epidemiological outcome. You know, we have some strains that spread quite well among the community, and others that we call them non-prosperous because they kind of didn't spread to the community as much as the other, but they are very genetically related. So it's like a good model to evaluate these kind of questions. We have some preliminary data, but still is too early to mention it, I think.
Okay, well, we'll look forward to those. So I'm noting there's a hand up from Brian. We'll take that, then Piyush, and then there's a question in the chat. So Brian, would you like to ask your question? Okay, he's got shy. Piyush, we'll try you again. Oh. Piyush, you go ahead.
Yes. Hi, Luciana. It's a great work.
Thank you.
I have an experimental question. How did you measure the ROS in mitochondria? I guess you showed a picture where you measured ROS in mitochondria, right?
We measure
reactive oxygen species.
Yeah, we measure it by seahorse, but not in mitochondria within the cells.
Okay. So you used seahorse method.
The seahorse, and we also complement the measurements by using a single cell method that is called CONICET, in which you can use different drugs to inhibit the different metabolic pathways, and you measure the incorporation of puramycin as a readout. It's like a readout of protein synthesis, but it has been really well demonstrated that it has a clear correlation with the bioenergetic status of the cell. So, and this can be evaluated by fax, so it's quite easy to adapt to any lab. And it has also the advantages that you can use fixed samples. You can use, you can use fixed samples and you can evaluate it complex samples. So you don't have to isolate different cell populations, and that's very important because when working with metabolism, just the mere isolation of the cells can have an impact on the ex vivo metabolic activity. So that way we try to use this this methodology, and especially we apply it for the characterization of the metabolic profile of the ex vivo monocytes.
Okay, great, thank you.
You're welcome.
Okay, so there was, I don't see Brian's hand anymore. There was a question in the chat from Sasha. She asked, have you tried this methodology with bowel fluid and compared it to
Yeah, we haven't. We really haven't. But I think it will be also problematic to do so because, you know, bowel is the problem that we have with bowel is that it's very affected depending on how much you dilute the sample. You know, sometimes to extract the sample, you have to incorporate saline to the patient, and then to talk about physiologically relevant concentrations, it's kind of a problem in that sense because it really depends on how much saline you add to the patients and how reproducible you are on getting the sample. That's why we consider that this sample, even when it is not representative of all TB patients, because we can also talk about pleural TB, it's a sample that is not diluted. So we got what it is within the pleural cavity. So.
Okay, thank you. So I'm just waiting to see if I see Mama having
hear
sound problems, but I'm able to hear and I'm not seeing other comments on that. So I think it's, yeah, others can hear.
Okay.
Okay, I'm just giving it a second or two for any other questions, but I'll just want to check in with Blakely because I know you like to do your photograph. Blakely, are you going to want to do that?